Thank you all for joining us this morning. My name is Daniel Paull. I am SVP of Discovery and Platform Development here at the New York Stem Cell Foundation. And so today's webinar is really a primer for everything you need to know and want to know, hopefully, about the promise of stem cells and stem cell research. And so I'm talking to you today from our laboratory here in New York City, where we are the New York Stem Cell Foundation. So JAX-NYSCF, as we call it, was established back in 2005, a little over 17 years ago, and during that time has grown enormously. And we now have a full-time lab here with over 100 different people working in a whole variety of ways, and we'll touch on some of that during the talk today. I'll just point out, thank you very much in advance for the questions that we've received. So if you submitted one, thank you. I'll do my best to try and address many of these throughout the talk, but as we go along, if you do have more questions, feel free to put them into the Q&A, and we'll try and address some of those with a question session at the end of this. So let's get going. There we go. So I think fundamentally we'll just start with this, and that is that we are not mice. And yet so much of research that is done today is done in things like rodent models. And these are great. Over the years, they've obviously delivered thousands of different medicines, and they've been a big part of that research and our understanding of disease, but ultimately they are not the human brain. And so while they're terrific and they do offer an enormous amount of research potential, they can't fully give us the full window into how a human disease develops and progresses over a patient's lifetime. And ultimately, it comes down to this sort of question, and that is that we cannot treat what we do not understand. And so if we're not understanding a disease and the way it is progressing, or even the way that a drug is affecting a disease, we really can't begin to move beyond it. And so what we really want and what we really need are windows into the human cells that are actually affected by this disease. But this is, of course, enormously difficult. If you've got a disease affecting the brain, like Alzheimer's or Parkinson's, or maybe diabetes affecting the pancreas, it is very difficult for us to obviously be able to take cells from a living individual. And so while we're enormously thankful to participants that donate things like their brain to brain banks, this is all post-mortem. And so by then, you're looking at the disease at a time when the disease has already progressed to its sort of very terminal phases, so to speak. And what we really want to be able to do is understand how a disease progresses over time, so that we can develop medicines that will allow us to intervene along the disease's progression. So the human body is made up of trillions of cells, and these obviously begin somewhere, and where they begin is at a stem cell. And so there's stem cells are a big term that is used in a whole variety of different ways. And looking at some of the questions that came in, it's clear that there's a lot of familiarity with the term stem cell. But just to point out a couple of key differences. In your body, as an adult, even as a child, there are stem cells in your body, but most of these have a specific sort of function. So things like a blood stem cell, or stem cells that you might find in your muscle, fat, bone, things like mesenchymal stem cells. These cells can only really reproduce the same kind of cell that they're already making, so to speak. So a blood cell can really only make a blood stem cell can really make only other types of blood cells. A blood stem cell, for example, cannot make a neuron, or it cannot make a piece of bone, or it cannot make a pancreas. The cells that can do that are cells that are termed pluripotent stem cells. And so these are cells that can really, truly make any cell type in the body. And classically, I think when people have thought of pluripotent stem cells, that has gone toward the embryonic stem cell. And for a long time, that was the model that stem cell researchers were using to do their research. And they were terrific models. They allowed us to make many different cell types, but ultimately they didn't really represent a given person or of a given disease, so to speak. And so what happened is that in 2008, 2007, 2008, a couple of landmark papers were published. And what it led to was the field of stem cell research really exploding. And that was because something named induced pluripotent stem cells came along. And so this work was discovered by a Japanese researcher who was working in San Francisco as well. His name was Shinya Yamanaka, and his work actually won the Nobel Prize in 2012. So 2007, this discovery happens, and I'll explain it in a second. 2008, we start seeing these cells really get put to use. And by 2012, the recognition that they were so important won him the Nobel Prize. And so what are induced pluripotent stem cells? Well, what they are is they are a representation of any given living person. So we can take a very small piece of skin, about the size of an apple seed, or a tube of blood, just like when you go to the doctors and have a blood draw. And we can take the cells either from that little bit of skin or from that blood, and we can reprogram them. So we change the way the cells are behaving using a genetic technique, and that allows us to revert those adult cells, those skin cells or those blood cells, back in time into a pluripotent stem cell. And this now opens up all sorts of different windows, because with these cells, they now have the genetics of the person from which they came. So your DNA, if I was to take a piece of a blood draw from myself, and I was to turn those cells into stem cells, those cells would have my DNA inside of them. And so that means that any characteristics of a disease that might progress would be captured within those cells as well. And because they're pluripotent, we can, of course, do all the things that we want to be doing with them. We can make all the different types of cells, whether it's a brain cell or a liver cell or a pancreatic cell. And the reason we can do this, again, almost goes back to those original animal models. We learned a lot of developmental biology and how things are developing over time. And so we've been able to apply that to human cells, so that we can make all these different types of cells that are ultimately affected in a given disease. And so where this leads us to is to having windows into diseases in just a truly unparalleled way. So things like Alzheimer's, schizophrenia, Parkinson's, we can make neurons from those cells. Things like osteoporosis, we can make bone and we can model things like that. Heart disease, diabetes, liver diseases. We now have the ability to collect samples from a patient that is affected by a disease, turn them back into stem cells, and then turn them into those cells that are affecting that person. And this really is this new paradigm, this new window into understanding how a disease progresses and how ultimately we can then test drugs and medicines in order to stop, halt, or even revert the progression of those diseases. So things like this, we can grow quite easily in a dish. You can see this beating away of these heart cells. And just like if you were to be in your house in the evening and maybe there's a loud noise and you get a spook and your heart starts beating faster because adrenaline or epinephrine is released into your system, the same way, if we give these cells some epinephrine, they'll start beating much faster as well. And we can see things like arrhythmias and things like long QT syndrome can all be seen in these kinds of cells. We can grow brain cells, all different kinds of brain cells, not just neurons, but more complex cells as well that we're beginning to realize have very major roles in a given disease. And we can grow beautiful cells that look like this. And these are cells that you find in your eye, and we'll come back to them a little bit later, but these are called retinal pigmented epithelial cells, and they provide a major role at the back of the eye to allow vision to happen. And so these stem cells, these induced pluripotent stem cells really are human avatars. They are your cells, a patient's cells that we use to study and treat that person's disease. And so really there are three main spokes that have come out of this ability to do this. One is disease research, one is drug discovery, and the other is cell replacement therapy, which I think is the natural one that most people go to when thinking about stem cell research or stem cell use. But today I'm gonna tell you a little bit about all three of these and how we're doing some of that, especially in collaboration with others, but I'll point out along the way where other groups as well are moving this kind of research forward. So I'll start with disease research and give you an example of how we're using stem cells here. So a complex disease, post-traumatic stress disorder. Conventionally, you might not think of this as something you could study with stem cells, but sure enough became something that we were able to look at. And so 1 in 11 people in their lifetime will be diagnosed with PTSD. And This obviously can be seen in a variety of different ways, whether it's just in natural life, but also in stressful environments like in the war zone and things like that. And PTSD has historically been studied using blood of affected individuals. And you get these snapshots in time of an individual and how their disease might be progressing or changing over time. But ultimately, PTSD is affecting the patient's brain. And it's really the effects of how the brain cells are responding that we might need to understand how a disease like PTSD happens and progresses. But inevitably, as humans, there's a lot of variation, ancestry, sex, various different things. Your DNA plays a major role in driving differences between any given individual. And so the way the disease affects people will be very unique from person to person. While a disease might have a similar bucket, so Parkinson's is a big term, for example, the way it progresses in different people will be very different from one to the next. And ultimately then as well, the drugs and the medicines that we have available for these kinds of diseases will affect and treat people in slightly different ways. And so we need to study large numbers of samples to better understand and treat these diseases. And here at JAX-NYSCF, we think that discovering new treatments for these diseases requires a lot of scientific convergence. We need to bring together groups from different areas in order to be able to treat these diseases. And so one way that we're doing that here is by developing something called the JAX-NYSCF Global Stem Cell Array. And this is a big operation that we've put together here, where we've brought together engineers, both software and hardware engineers. We've brought together computer scientists and data scientists, and then biologists as well, all working together on a very advanced robotic platform in order to be able to do this kind of research at very large scale. It's quite typical for a lot of research to be done with small numbers of samples, maybe 3 or 5 or 10 different individuals, but that doesn't really capture the diversity that you might see in a disease. And so one of the ways we chose to try to tackle this was to develop these very large automated platforms that can do everything that I used to do by hand, I can now do on this big robotic platform here. So it can feed my cells for me, it can turn those stem cells into those affected cells, and then ultimately I can use these kinds of robots to test all sorts of different medicines at the same time across different individuals and different cells. And so this kind of robotic platform is what we use to try to understand PTSD. And so here we teamed up with teams at Mount Sinai here in New York and a team from the Veterans Affairs Department. And we collected material from veterans that had served overseas. So all of the subjects in this research had served overseas. 20 of them came back diagnosed with PTSD, and 20 who had seen similar traumatic events came back unaffected clinically. So we collected skin biopsies, those little pieces of apple seed-sized skin, and turned the cells from that into stem cells, and then we turned those into neurons. So we had all these different cells growing at the same time on our robots from these different individuals. And then what we wanted to do and try to understand is, if we stress these cells, can we see a difference in the way that maybe the PTSD-affected veterans respond? And so mimicking what happens in the body, where your body releases hormones that cause a lot of stress, we've exposed these cells to something like hydrocortisone and looked to see if there was a stress response. And sure enough, what we were able to see in a very simplified way here is that while the controls sort of didn't respond too much to this induced stress, we could see this very big increase in cellular response to stress in the PTSD affected veterans. And we really could see very big differences between them. And so where does this leave us? This leaves us in a place now where you have a model of a disease like PTSD that we can now apply drugs and compounds to see if we can dampen that response in those elevated stress levels. We've done a whole lot of different research in this way across different diseases. We've worked with Harvard and Rush University to replicate Alzheimer's disease brains in the lab, so growing cells from Alzheimer's patients in the dish and looking at the way that those cells change over time. We've been looking at the role of inflammation in multiple sclerosis, and this is big collaborative work with teams at NYU and Johns Hopkins as well. With the National Institute of Health and the ex-director of the NIH, Francis Collins, we're looking at risk factors that affect type 2 diabetes. And we've been using very cutting-edge research with teams from Google to look at how patient cell, whether we can identify differences in patient cells and when those patients suffer from a disease like Parkinson's. And there's all sorts of webinars that we've had around these different topics over time, and there'll be some links in the chat if you'd like to go and dig a little deeper into some of these. So one of the other major areas that we're trying to do this kind of research is in drug discovery and drug development. And really what ultimately you want to get to in any kind of research is something like this, where you have lots of different people represented in your studies. So you have lots of different cell lines from all of these individuals, and you want a way to tell the difference between the cells from the person with the disease compared to the person's cells that don't have the disease. And then you want to ask one more question. If I give a medicine to those disease cells, will they appear healthy again in the presence of that drug medicine? And so if you think about something like Parkinson's, it is a very complicated disease. And what we asked in this particular example here, what you see in front of you here are lots of different squares, and inside each square is effectively a different person's cells. And so what you're seeing are cells from 60 different people in this picture, and about half of them have Parkinson's and half of them do not. And I have stared at these images. I have tried to come up with reasons why they are different. And if you were just to stare and look and look and look, it would be very difficult, nigh on impossible, to find any differences between them. These are just skin cells at this time. This isn't even stem cells here. You might see that some have less and some have more. That's a given, but fundamentally they look very similar. And so we wanted to come up with interesting ways and novel ways of detecting differences between these. And so what we did is turn to artificial intelligence, AI. It's obviously become the big term in our lives and is changing the way we do lots of different things, to ask, can we develop very, very sensitive ways of telling the difference between these cells? And where that led us to was to build models that at first, the data, these cells would all look the same. We couldn't really tell the difference. But as we refined the models over time, what we were ultimately able to do was really separate out and be able to tell the difference using these deep learning artificial intelligence models, the difference between healthy cells from healthy individuals, and the cells from Parkinson's patients, which is just fascinating. And ultimately, it leads you to a point like this. If we have these two different buckets, like I've just shown you, these two different groups, can we apply the drugs onto those cells and do this drug testing in the dish so that we're maybe able to shift the patient cells so that they look much more like the healthy controls? And in that, maybe you are finding new medicines that can treat a disease. So we've been doing this a little bit in Parkinson's, as I've mentioned, but we've also been doing this a lot in rare diseases and in a few other areas. One rare disease that we have been looking at is a disease called infantile neuroaxonal dystrophy. It is an ultra-rare disease affecting only a few hundred children in the world, but these children are affected very early in life and have a number of neurological impacts from this. So what we set out to do was build that kind of model I have just shown you using artificial intelligence. And then we screened 3,000 drugs that the FDA has approved over the years to see if they could be repurposed to be used in treating this disease. And where we are now is that we've identified a number of compounds that look very promising. And so we're now narrowing down those drugs to see which could be potentially clinically beneficial. And so finally, this brings us to cell replacement therapy. How could we replace the cells affected by a disease so that we could either prevent or rescue the effects of a given disease? And the example I'm going to show you is age-related macular degeneration. This is one of the world's leading causes of blindness, affecting hundreds of millions of people already, and just continuing to rise. It's associated with age, obviously, as the name suggests, but can occur in a couple of different ways. And what happens is, as I was talking just then, hopefully you noticed that this photo became quite affected. And this is representation of what happens in a disease like macular degeneration. And while there are some treatments, they really only slow the progression of the disease. They don't ultimately treat it or in fact, rescue it. And what is happening in this disease is this is what the retina looks like, the back of the eye, and you'll see that there's this patch here missing. And what's happened here is that a group of cells called retinal pigmented epithelial cells have been lost. And these cells die with age. And as they begin to die and are lost over time, the photoreceptors that actually detect light and allow you to see also can no longer survive without that protective layer of those cells. And so this has led us to begin a study here, building on the back of other people's work that have done this before in some embryonic stem cell work. But here we're taking a very personalized approach to treating this disease. And so the plan is, and already this work is underway, where we are collecting samples from patients affected by this disease. We've been collecting blood in this example, taking those blood cells and turning them into stem cells, turning those stem cells into those affected eye cells that I was just describing, those ones that are lost with age. And you grow these, they are relatively straightforward to grow, and you put them onto a patch. And this patch can then be prepared for surgery to be transplanted right back onto this area where they are lost here. And this will complete the circle and go back to the same patient. So a very personalized medicine where it is the same person's cells being taken through this entire process to have these cells transplanted back into them and hopefully either rescue or prevent the progression of this disease. And this is work we're doing in collaboration with Columbia University, and the trial will hopefully be beginning next year. Here in New York, we've had to build very special facilities to be able to grow these cells in ultra clean conditions so that they can be used for these kinds of clinical applications. So hopefully we go from this to this and allow these patients to see once again. And there's a whole heap of different clinical trials underway for a whole variety of different diseases. And we're just going to see this continue to grow and grow and grow over years. So Parkinson's, there's been a very exciting trial started here in New York by a company called Blue Rock Therapeutics that have been transplanting the dopamine producing cells affected in Parkinson's disease back into the brains of patients. And those trials are only about a year into them, but already very exciting. And similar trials are underway in Europe with Lund University and some folks at Cambridge as well. In the diabetes space, there's a lot of interest about being able to transplant insulin-producing cells back into the body, and there's some fascinating work being led by Vertex in this space, but again, a whole heap of different companies sort of just on the precipice of being able to do their own trials in this way. And then a variety of other cell types being produced by companies like Fate Therapeutics and Heartseed in order to be able to transplant in cells in a variety of ways to treat many different diseases. In all, there's over 120 registered pluripotent stem cell studies underway at the moment. So just incredible to see how this field has grown over the past decade or so. And so I'll summarize with just a little bit about JAX-NYSCF and sort of our role in this sort of ecosystem and the stem cell space. So as I mentioned, I'm talking to you today from our research institute where we do a lot of the work I've spoken about today. But as a foundation, we have a driving mission, and that is to accelerate cures for these major diseases of our time using stem cell research. So while we can do that ourselves here at the Institute, we also do it in a few other ways. And one of those is education and outreach and webinars and seminars like this, but also helping to build communities in the stem cell space. And we have funded hundreds of researchers around the world, and we bring them together in order to be able to meet and explore ideas. And it's been quite exciting, different companies and different collaborations that have come out of bringing individuals that might not otherwise have met together in this way. And here at the Institute, it's become a pretty unique model of research. We are completely independent. We have collaborations with many institutes, as I've mentioned, but we are ourselves independent, and we are privately funded. We're a non-profit doing this work to move stem cell research forward. And so I've spoken about some of the technologies that we've pioneered here. There's many, many more that we again are in webinars and things that you can go away and hear about. I've mentioned we collaborate with many different institutions around the world. And one of the big ones that we also do is provide cells to researchers. So getting access to material to study a disease can be very challenging as a scientist. And so one of the things here is that we've built a biobank of cell lines, and we ship those around the world every day to researchers to be able to do their work as well. And so, as I said, on staff here, it's over 120 staff spanning all sorts of areas from biology to computer science to hardware engineering. And here at the Institute, we study, as I've sort of mentioned throughout the course of this, many of these diseases we've touched on today. So things like Alzheimer's and Parkinson's, MS, autism, and a variety of neuropsychiatric disorders. We also have teams innately working in areas like women's reproductive cancer, diabetes, rare disease, and retinal diseases. And so that marks the end of this portion of the webinar, and we'll open it up for questions and see where we go from here.
Thanks, Dan, for that amazing talk. So we had a number of questions that came in around the macular degeneration work that you talked about. So you mentioned it briefly, but can you talk about what that timeline looks like and what the process is like of taking that stem cell patch that we have and turning that into a therapy that can reach the masses?
Yeah. So it, as with any trial, clinical trials start very small. Really, at the beginning of a trial, you're looking for safety and really, if you see what we call efficacy or benefit from it, that's really a secondary benefit at the very earliest stages. And so that's sort of where we are now. It's a small number of patient samples that are being turned into stem cells, being turned into these eye cells and put on these patches. But clearly the goal and the need is to make this accessible to as many people as you can over time. And so there's all sorts of ways that this will shift over time. There's going to be big technology leaps in things like automation and robotics that allow us to continue to scale these things so that they can become accessible. And there's a number of approaches that we're looking at as well, like can you camouflage cells so that I didn't really talk on it today. One of the issues with using things like embryonic stem cells is that they aren't your own cells, and so it would be akin to getting a transplant where you have to go on immune suppression to prevent rejection of the tissue. But there are ways, and people are exploring them, to camouflage the cells to prevent them being rejected in that way, and maybe that's another approach that over time becomes an approach to scaling this.
There's also a number of questions around stem cell therapies for neurological diseases, including the Parkinson's work. But there's also some questions about, a lot of different types of therapies that people are interested in trying. Without getting into specifics, can you talk about sort of the need for clinical trials and sort of some of the issues that are out there with what we might call stem cell tourism?
Yeah. So as we sort of said in the talk, the field has moved in just enormous leaps over the past 10, 20 years or so. But it typically takes a medicine or a drug well over 10 years to become a treatment. And so we're really only at the beginning of this using things like clinical trials. And as I mentioned, the importance of clinical trials, especially at the beginning, is safety. That's why they start very small, because you want to make sure that you're not putting a cell back into a patient that would actually make things worse or cause some kind of adverse reaction. So they start small for a very specific reason. What we have seen over the years, things that the field of stem cell research has battled with is this concept of stem cell tourism, where large, quite elaborate claims have been made about the use and the utility of things like these adult stem cells to treat a number of diseases. And there haven't been the kinds of rigorous studies that have been then peer reviewed and gone through the right channels to know whether they're actually beneficial. It's a lot of sort of anecdotal evidence or things that may not even be real. And so I think I would just caution individuals that have been thinking about exploring it. There's a lot of great information on our website, and there's another organization called ISSCR, the International Society for Stem Cell Research, and you can go both to us and to them in order to find more information about these kinds of issues that come up in this space. I think, like I said, we're gonna see great research reach patients very soon, and these things do unfortunately just take a little bit of time to be done correctly.
So we're at the time. So with one final question, what excites you most about the field, and where do you see the future? We're still in the early days of stem cell research. I mean, you mentioned it was just 2007 when this cell type was first able to be made. What excites you, and what does the future look like, down the road?
Yeah, it is. Yeah, but we're really right at the beginning still. 20 years might sound like a long time. It's really not. And so we're really right at the beginning of this. And our ability to make cells that are affected by in a given disease is only getting better. There are still some cell types that we can't make that we don't know how to make yet. There are some cell types where it takes just a very, very long time to make them. And so to have a clinical benefit from them is still quite challenging. And so there are going to be technology leaps that will allow us to create these cells quicker, the right cells a little bit better, so that it opens up this as a therapy to more and more different diseases. And then I think tied to that is, and something we think about a lot, is making this kind of work accessible. So as I said, cell therapy is definitely a challenging one from how you make enough cells to treat enough patients. But the impact it will have in drug discovery and finding or repurposing medicines is going to be enormous. And we're just beginning to see big pharmaceutical companies really jump on board with this to improve their models that they're using, to move away from those mouse models and some of the cell lines that are being used right now. And once they jump on board, and like I said, that's really just ramping up at the moment, this is where it will really begin to take off, and we'll see therapies that you take at home really, really become possible because of stem cell research.
Well, thank you, Dan. And thanks everyone for joining today's webinar. There was a few questions about if we'll be sending this out. Yes, this will be on our website, on YouTube in the coming days, and we'll be make sure to share that with everyone. So thanks a lot for joining.
Thanks, everyone.